So today, my topic will be silicon and abiotic stress in plants, current knowledge and future research needs as requested by the organizing committee. My talk will be composed of the 4 parts. We know silicon is the second most abundant element of oxygen on the surface of the Earth's crust and in the soil. So although it is not an essential element for high plants, it is a beneficial, agronomically essential element. So as pointed by Professor Epstein, silicon is one of the quasi-essential elements. All plants growing in the soil, can absorb silicon, but, silicon content in plants, ranges from 0.1 to 10% silicon on a dry weight basis. So it is dependent mainly, on plant species and external silicon concentration. So based on silicon concentration in plants, we can divide the plants into the following 3 types. One is silicon accumulator, for example, rice and sugarcane. A second is intermediate type, such as cucumber, sunflower, and soybean, the dicots. And the third is excluded species, for example, tomato, fava bean. These plants exclude silicon from uptake. But this is only based on the silicon content in plant tissues. Rice is a silicon accumulating plant species. Silicon is required by rice heavily. Silicon concentration in rice is bigger than the total NPK. So if the silicon content in rice straw is less than 10%, that means silicon deficient occurs in plants. In Japan, their work started in 1952, and the silicon is recommended by Japanese government by law in 1955. And in other countries, Southeast Asia countries and districts, for example, Republic of Korea, Thailand, Philippines, Malaysia, and Chinese Taiwan, silicon application is a common practice in agriculture. So in Chinese mainland, we started to do the experiments as early as in 1950s. Professors and scientists working in the institution and the colleges, Institute of Soil Science, Chinese Academy of Science, and also Zhejiang Agricultural Universities. We have done a lot of experiments on a large scale. Many crops could respond positively to application of silicon. The yield increase in rice, wheat, sugarcane, maize, cotton, cucumber, tomato, strawberry, and watermelon could be very high, at least on the average 10%. Many soils in China are silicon deficient. At least 300,000 hectares of paddy soil is silicon deficient. So the potential market in China is huge, about 40 million tons per year. So, but we need developing this market. My topic is mainly silicon and abiotic stress. We think silicon is an anti-stress element for plants. We have 3 categories here. One is abiotic stress. Second is biotic stress, as reported by Danoff. So for abiotic stress, we have physical stress, such as waterlogging, drought, freezing, or cold chilling, and extremely high temperature UV radiation, soil compaction or heartburn, etc. And for chemical stresses, we have aluminum toxicity. This has been extensively studied over the last 3 decades, but recently less information has been available aluminum and silicon interactions. But cadmium, lead, arsenic, chromium, zinc, copper, manganese, and iron toxicity have been extensively investigated with silicon. Another one is nutrient imbalances, for example, phosphorus deficiency, nitrogen excess, potassium deficiency, low soil organic matter and boron toxicity, and also salt stress and pH. It is too low or too high. My talk will be focused on metal tolerance and salt stress. So we have another category, I call it abiotic stress. The typical example is lodging in cereal crops. It is neither abiotic stress nor biotic stress, but abiotic stress maybe. We did the experiments with maize. Silicon could significantly increase maize tolerance to cadmium toxicity and improve plant growth. You can see very distinct difference between the cadmium treatment and cadmium plus silicon treatment. If your experiment just like this, then your experiments are very successful. This is interaction of silicon with zinc and manganese toxicity in rice. You see, silicon could enhance resistance to excess zinc and manganese toxicity in rice. You can see very clear the root morphology. Also very clear, difference between zinc treatment and zinc plus silicon treatment. This is two different cultivars. This is resistance. This is sensitive cultivars. From these figures, you can see, silicon could significantly improve the growth of rice exposed to excess zinc and manganese stress. So very clear. So now let's talk about silicon and salt stress. So as early as in 1996, we published a paper in Journal of Plant Nutrition reporting the findings for silicon and salt stress in barley. You can see this is the salt tolerant cultivar, and this is the sensitive cultivar. You see the performance and barley with silicon and without silicon is different. So you can see this is the roots. Okay, so the first time we proposed that silicon could increase salt tolerance through mediating antioxidant defense capacity. We proposed this hypothesis in 1999 and 2003. After publishing of these papers have been frequently cited. After we published this paper in these journals, in my group we have done the further research, and some other colleagues all over the world did similar experiments, but with dry crops, for example, cucumber and tomato. They confirmed our hypothesis. So many other works have been reported in the international journals. I will give the information later. So we further look at plasma membrane and tonoplast protein APTs and protein pyrophosphate activity. Why? Because we observed that silicon reduced sodium but increased the potassium uptake in salt-stressed barley. Silicon could increase root plasma membrane and tonoplast protein ATPs and protein PPs activity in barley and salt stress. And also plasma membrane and tonoplast fluidity of roots and leaves of barley and salt stress could be significantly increased by addition of silicon. As I give you here, you can see very clear the root tonoplast protein ATPs activity was significantly increased the silicon in barley and salt stress. And this is plasma membrane, the similar results. So root and leaf plasma membrane protein ATPs activities were significantly increased by silicon in barley and salt stress. We investigated into the index of unsaturated fatty acid, IUFA, this parameter, and the fluidity of tonoplast vesicle in salt-stressed barley roots. You can see this parameter was significantly decreased by silicon compared with salt treatment only. And the fluidity for tonoplast vesicles was decreased by silicon. Based on the publications in the international journals, most of these publications, most of the work was done in my lab. We just summarize the interactions between silicon and salt stress in plants. We have osmotic stress, nutrient imbalance, and ionic sodium toxicity in plants under salt stress. Silicon uptake by plants Silicon could increase root activity, then enhances mineral uptake, then could inhibit nutrient imbalances. Then silicon could decrease salt toxicity. As we know, silicon could inhibit transpiration. Then osmotic stress could be reduced. Then you see this is another pathway. And the third is, as we discussed about, silicon could increase ATPs and PPs activity in PM and tonoplasts, then increase potassium uptake, decrease sodium uptake, then reduce ionic sodium toxicity. So as we can see from the literature, silicon could also enhance the strength of cell walls by enhancing lignification and suberization in plants. So then it could decrease oxidative damage by increasing antioxidant defense activity and reduce membrane permeability and improves its structure and stability. So silicon could significantly decrease salt toxicity in plants. You can see many publications in silicon and abiotic stress in relation to antioxidant defense activity. So boron toxicity stress and cadmium stress in rice, and in the pak choi and freezing stress in wheat, drought stress in wheat, zinc toxicity in rice, and a manganese toxicity in coffee, cucumber, and in rice. So you see, it's very clear, silicon could increase the antioxidant defense activity in pak choi and cadmium stress. We use, this native page methodology to analyze SOD and CAT isoforms profiles in pak choi treated with cadmium and silicon. You can see this is the sensitive cultivar, this is tolerant cultivar, and it's very clear, treatment with silicon could increase the intensity of SOD and CAT isoforms in cadmium-stressed pak choi compared with the corresponding cadmium treatment alone. We also use histochemical methods to determine membrane integrity and damage. It's very clear that silicon could increase this membrane integrity and reduce damage by reducing lipid peroxidation. Cadmium-treated roots will stand to different extents, and under the higher cadmium treatment, the roots will stand extensively, but the roots become lighter straining in the cadmium plus silicon treatment compared with the cadmium treatment alone. The similar findings with silicon and cadmium interactions in rice, you can see very clear. So the rice roots will much less stand in the silicon plus zinc treatment compared with zinc treatment only in rice. Frack and his colleague in Germany found silicon could increase suberization of the endodermal cell layers of rice roots. It's very clear, you can see here. Silicon could reduce radical oxygen and oxidation power, thus increasing suberization of exodermis and lignification of sclerenchyma. So this change indicated by silicon supply was related to an increased transcription of genes responsible for suberin and lignin synthesis. It's clear, silicon could increase antioxidant enzyme activity and oxidant concentration, thus reducing oxidative damage in plants under abiotic stress. For example, salt stress, drought, and freezing stress, excess cadmium, boron, manganese, and zinc stress. So we propose that silicon enhanced antioxidant defense activity is a universal mechanism for silicon enhanced resistance to abiotic stress. So silicon could also increase tolerance to abiotic stress and improve plant growth through increasing metal tolerance via less metals transported from roots to shoots. So you can see here, it's very clear. Cadmium in shoots decreased and in roots increased by addition of silicon and cadmium stress. This is also in zinc. You can see very clear. Zinc concentration in rice roots was significantly increased in the zinc plus silicon treatment, but was significantly decreased as compared with the high zinc treatment alone. So this is the manganese case. You can see here, this is sensitive cultivar, this tolerant cultivar. This is manganese treatment. The concentration in shoots similar, but in roots much higher for manganese plus silicon treatment. But for this tolerant cultivar, manganese concentration in shoots decreased a lot in the treatment with manganese plus silicon treatment compared with manganese treatment only. In roots, also decreased very much. In the sensitive cultivar, solution to root manganese transport was restricted. But in the tolerant cultivar, root to shoot manganese transport was inhibited. So we just use synchrotron radiation X-ray fluorescence to study relative manganese content in rice root tissues. You can see very clear, less manganese was found in the tolerant cultivar than in the sensitive cultivar too. Less manganese was found in the manganese plus silicon treatment than in the manganese treatment only. So similar results were found with zinc. So we can summarize all these findings as follows. You can see here for metal tolerance, we have external mechanism and internal mechanism. For external mechanism, immobilize heavy metals in media or metals and silicon co-deposition in solution. But for this internal mechanism, maybe we have different pathways. For example, metals and silicon co-deposition in plant roots. Silicon could reduce metal root to shoot transport, and silicon could enhance metal tolerance via more metals bound to the cell walls. And another most important factor is if after silicon uptake by roots, silicon could enhance root growth and strength of cell walls by mediating this lignification and suberization, then increase tolerance, decrease oxidative damage by increasing this antioxidant defense activity and suppressing the production of free radical oxygen. So now let's look at some molecular studies on silicon-enhanced resistance to heavy metal stress. We just use high throughput sequencing technology to identify genes which express differentially in silicon-amended and non-silicon-amended rice exposed to excess zinc and manganese. You can see here for zinc, about 4,115 genes were upregulated and 861 downregulated for this silicon plus zinc and zinc treatment. And you can see here, for manganese, we have 647 genes upregulated and 892 genes downregulated. So, based on this data, we just use quantitative real-time PCR to analyze the differential expression of genes related to photosynthesis in rice grown with or without silicon and manganese toxicity. So you can see silicon added could significantly enhance relative gene expression of these genes. These genes are related to photosynthesis, so that's enhancing resistance to zinc toxicity. Okay, now in conclusion, let's summarize. Silicon is not inert, but could enhance plant resistance to varying forms of abiotic stress, for example, salt, heavy metal stress, via metabolically active processes. Second, silicon-enhanced antioxidant defense activity is a universal mechanism for silicon-enhanced resistance of both silicon accumulators, such as rice, and non-silicon accumulating plant species, such as pak choi, to abiotic stress. So heavy metal toxicity could be alleviated by silicon via more metals bound to cell walls and less root to shoot transport. Silicon enhances salt and heavy metal tolerance by mediating membrane integrity and stability. Silicon could enhance resistance to metal toxicity through regulating expression genes responsible for metabolic processes such as photosynthesis and oxidative stress. So silicon is a unique beneficial element for high plants. Its essentiality, however, needs clarifying by accumulating more direct, convincing evidence at molecular level. Further research should be cloning and identification and functional analysis of silicon genes responsible for silicon uptake in dicots such as sunflower and cucumber, and the molecular aspects of silicon-enhanced resistance to abiotic stress also needed. So more research work is needed on interactions of silicon with nitrogen, phosphorus, and potassium in high plants, which should have been extensively studied. More lab and field work is needed to improve the growth, yield, and quality of crops of agriculture and horticulture importance under stressful environments. So these are parts of my colleagues, and thanks to the fundings provided for our research. Thank you very much.